Hello, my name is Alanys. I am from Mexico and I am studying in UVU Biology University. My major is biotechnology. Not only a student, but I also work as a teaching assistant, and as well as that, I do research. When I was in middle school, I discovered that I really like to spend my time in the laboratory because it's really fun and I really like to wear my lab coat. So it was my favorite part of classes to go into the lab and wear my lab coat. Science could look challenging, mostly if you are a woman. It could look like maybe you don't belong there, but I feel like it's a special place, it's a beautiful place, the laboratory, and as women we belong where we want to be, and just the laboratory is so much fun. So today we'll be performing a purification. We're living through a pandemic, so the only way to fix it is through vaccination. So our goal for today is to have a pure RNA vaccine. RNA is the information that viruses use inside the body to reproduce. So with this special RNA in the vaccine, it can get into the body and not reproduce the virus, but provide protection. Biotechnology companies are able to produce special RNA that we use in the vaccines, but unfortunately, it has contaminants. These contaminants are harmful to the human body. So we as a biotechnologists are super cool, and we are able to get rid of those contaminants using purification. In this column, the contaminant is going to be behind, and we have the RNA purified. Once the RNA is pure, we can inject that into a person, in this way ending the pandemic. So for today, we're going to be showing you this little step, which is leaving the contaminant behind and purifying the RNA. So right now, we're going to start the purification, and we have right here the RNA and the red contaminant. So we'll put it through the column to purify it. This column contains some special beads that will retain the contaminant on the top, and then the pure RNA will flow through it. So now my fellow biotechnology scientist will help me show you how to put the RNA and the contaminant in. So as you can see, it's going into the beads.
So at the moment it's just a mixture of the blue color, so the blue RNA and the red contaminants. So you cannot see them really separated at the moment because again it's a mixture. They are all together. Now I'm going to push using this pump here, the syringe, a liquid, and then you should be able to see the beginning of the separation. So you can kind of already see the blue front here and the red staying up.
So now you can see a little more blue, and it's going faster than the red.
From the blue, then you can see like a mixture after, and then the red in the back. So the red is the contaminants are retained, while the RNA is already kind of pure here, because there is no more overlap between the red and the blue. So you see really the blue.
So blue starting to come to the end. And remember, the RNA is the ones that we want. Blue RNA is almost at the end, and the contaminants are still a little behind. And we are collecting right here the blue RNA, purified blue RNA.
So here it's yellow because it's kind of a mixture between blue and red.
And it's getting darker. That means that there's more contaminants there.
So means here that you have a pure sample right there and here, where it's kind of very clearly where it is blue. So this is the blue thing here is actually what you can inject
safely
in in the person.
We hope you enjoyed this video on how vaccines are produced and purified, and also how biotech is important and applied in our daily lives. Thank you so much for watching, and we hope to see you in the lab one day. Have a wonderful day.